Hey everyone, this is Chris Thompson of the Purdue University iGEM team in collaboration with the 2013 Copenhagen iGEM team, creating the Bricks of Knowledge videos. And this short tutorial is going to explain how to use the iGEM registry online. And the iGEM registry is basically a big catalog of the different DNA parts that iGEM teams use to complete their projects. So we're going to show you how to use it. So to start off, you want to actually open the iGEM registry. So for this, you can go to Google, just type in iGEM, and the main page should be the first thing that pops up. And this is the main iGEM page. If you need to know anything about the competition or anything like that, this is the place to do it. But today we're worried about the registry. So it's the third tab from the left. Click it there. This is the registry main page. Featured on the registry, a little blurb about what it is. But we'll show you kind of how to navigate this page. So easiest way is the navigation bar. First tab here is just the iGEM tab, your quick links about the competition, about iGEM, what the competition is completely about. You find everything there. Then the tools tab, anything you need to know about the wiki, the general about iGEM, add a part, request a part, the community, find everything there. The catalog, and this is one of the most important tabs in the navigation bar, and we'll definitely get back to it later. Browse the catalog, browse by type, other, we'll get back to that later of actually browsing the catalog. And then the repository, the DNA distributions, and we'll actually talk about this real quick as a side note. We're going to show you how to search the online registry for parts. This is a really easy way to search through the actual iGEM distribution. So every year, iGEM headquarters sends every team a giant catalog of parts that they can then use in their lab. So instead of looking online, you can actually just look through these parts, like this list. So in each distribution, there's a bunch of different kit plates. So let's say kit plate 1. You can actually get an Excel file for every kit plate. So instead of going online and looking through parts and trying to see if they're in the registry, you can actually just download the Excel files, look through the Excel files on Microsoft Excel, and then find all of the information you need on there. You just press get an Excel file for this plate, and you can do it for every plate. So that's how you do it not using the actual website. So we wanted to show you that real quick. We'll go back to the repository tab, and all of those for all the previous distributions are there as well. And then how to use the samples, how to send samples, request a sample, stuff like that. Then the assembly. This is really great place to learn about general assembly, the standards, the 3A kits, the methods. Again, really good learning tab. Protocol is very similar. Registry tested protocols, so making competent cells, transformation. Figuring out what your school needs to do iGEM. This is a really good place to do it. The community, the assembly, everything like that. And then learn, it's pretty obvious, about, again, help system. One thing the registry does really well is try to get as much information to you as possible to help you learn how to do everything. So this is where you can find all of that. So, like I said, this is a tutorial on how to find a part in the registry. So we'll go to catalog. We'll go browse the catalog. And this is the way. We'll describe a few different ways to find a part. But this first way, let's say I'm looking for a part for my project that works in E. coli, is a promoter, and is a constitutive promoter. So knowing that information, we'll go to the catalog tab and press browse the catalog. So here you'll get a page where it separates the different parts by through different ways. So right here you have browse parts by type, another browse parts by type, browse parts by function, browse parts by chassis, browse parts by the standard, browse parts by the contributor. There's a lot of different ways you can find the parts. But with the information we had that I said, we want a promoter, constitutive, and E. coli, we'll just start with type. And this is, I think, the easiest way to do it sometimes. So we know we wanted a promoter, so we'll just go in, click promoter. You'll be brought to the promoter page. It's kind of the main page. It describes what it is, if you need to learn how to design it, any help you need with the promoter, and anything like that. But we already know everything about promoters because, we're smart. So we'll go to promoter catalog. This is a way to find the different types of promoters within promoters. So again, it lists it two different ways. You can browse it by function, which is the most common way, and really exists on every type of part. So we're only going to show you how to search through promoters. But this method and these methods should be applicable to all the different types of parts. So then we'll go through, there's another way, browse by regulation RNA polymerase. And this is, I think, a little bit unique to promoters. It's a little table. It's actually really easy to use. On the x-axis up here, you have the different types of chassis, so E. coli, Bacillus subtilis, miscellaneous prokaryotic, and all of those. On the y-axis here, you have the type of regulation, so positive, constitutive, negative, multiple. So easily using this table, which again I'm pretty sure is unique to promoters, or at least isn't on every part page. So we said we wanted an E. coli promoter and we wanted it to be constitutive. So we find the little table here. It says 63, so we'll open that in a new tab. And it'll take us to this page, where it just lists all of the E. coli constitutive promoters. Very easy, and we'll show you more on that in a second. But like I said, that's a little bit unique to promoters. So we can also just browse by function. So we wanted a constitutive promoter, so it's the first one. That's pretty easy. But there's also, the other different types of parts, or promoters. So then we bring up this page. So like I said, both of these methods and both of the links I just clicked are an easy way to find constitutive promoters. But the difference is, since we use the table, this list that we opened through the table is already kind of smaller because it only has constitutive E. coli promoters. And over here, it lists E. coli promoters, subtilis promoters. So this list is already a bit narrowed down, the one that we opened with the table. But either way, it's the same list, it's just longer or shorter. You can still find the same information. So we'll go to the list we opened with a browse by type, because that's on every single part page. So when we scroll down, and we wanted a constitutive promoter. So we'll kind of explain the tables here. This little symbol right there is the registry star. So if it has a registry star, or if it has multiple registry stars, that means it's been proven to work and people like it, it's been highly rated, and it gets a star. So a star is always a good thing. And this little green box with a W means it's been confirmed that it works. So it does work and it is available, or at least just works. So then you click a link and it doesn't actually bring you to a new page yet. It brings you to this little box to where you get the little symbol, you get the name, you get the description right here, how many uses it's been, and when it was submitted to the registry. So you can kind of easily compare on the same page without opening up 30 different tabs. Like let's say this is 2006, 59 uses, and it's available. This one, 58 uses, does have it available. So we'll look at this one. So you open this up, you actually click this link up here, which we'll go ahead and click. And it'll take you to the part page, which is kind of one of your final destinations for this. And since this part has been used 59 times, it's a really good part. It has a lot of information here. This isn't always what you'll see. A lot of times you'll open up this part page and there'll be no data, no information, and it'll barely have the sequence on there. So try to find parts, obviously, that have a lot of information here, because those are the easiest to use because you already know a lot about it. So this part has a picture of it being used. It has some DNA information, has some variant RFP information. There's a really good description here of all of the different parts of this. It has a plasmid map. And then, like I said, this information won't always be on there, but hopefully this information down here always will be. So you can open up single-stranded and you can actually see the different, the actual sequence for this part. You can also press this button that says Get Part Sequence, which you can actually copy and paste the sequence to put in another web page, or maybe you need to put it in Word, who knows. You go back, and then you actually press this button, View Plasmid, which will open up the actual plasmid that this was used on. Very, very helpful, a lot of good information here. You can even change what type of plasmid it's on, which is awesome. That's how you use that. So then you decide, by looking through all this stuff, okay, I want to use this part in my lab. So what do I do from here? Well, that's where this little table on the right becomes your best friend. So this contains a bunch of information of how to actually use this part. So you can see it was released by headquarters in 2013. It's in stock, which is always a great thing to see. Like we said, registry star, uses, and then twins. Twins are just parts that are very, very similar, but maybe have been updated, or this one was the most updated version. And then the Get This Part link. And this is, again, your best friend. This is If you don't learn anything from this tutorial, that's the button you need to learn how to find. Because this will tell you how to actually get to that part with the distribution you were sent. So you can look, I said, okay, we have the 2013 distribution. We can find out where it is in that. It's on kit plate 5, well 18G. It's in this backbone, and the sequencing's been confirmed. So then you can go to your lab, look at kit plate 5, find well 18G, resuspend the DNA, and use it. And I'm sure that'll be covered in different bricks of knowledge, but that's how you find everything. So that's awesome. So that was a bit long explanation how to find a part. So there are other methods too. So we'll go back to the main page. So let's say I'm looking for a part. I'm just looking for the RFP part. I don't know anything about, whether it's E. coli or anything specific. I just know I'm looking for RFP, which is red fluorescent protein. So let's try to use this little search bar up here. And this is the place where it gets a little bit unfortunate because the search algorithm on the iGEM website isn't that great. It's okay, but I'll show an example. Let's search RFP. And then instead of taking you to the actual RFP part by itself, it lists a bunch of pages that have RFP in the text somewhere, which is good. That's one way of searching, but that's not really what we want. And even then, if it had RFP in the text, it would at least show the RFP part, but it doesn't. So what we'll do instead, there's a little triangle next to this, and it brings up this little drop-down menu. It said, enter the text or part you wish to find. So we'll type in RFP again. And then it has these handy little buttons. Click to search using Google, using the Google algorithms, because iGEM realized their algorithm isn't the best. It's not the best way to search it. So let's search using Google, which is, the best search engine. So instead of searching through the registry itself, we'll search the registry using Google. And then right here, the actual RFP part still isn't the first thing listed, but it's second on the list, so it's pretty close. And that just might take looking through the different results and finding the one you want. So we see this, we found the part, we can use it. Again, we can press get this part and find where it is. But let's say we know more than we have before. Again, this is everything there. So let's say, well, I actually do know the part number. How do I search using that? Well, you can go up here, and the actual usage of this toolbar or this search bar, so you can actually type in the name. So we know that we just found out that the name for RFP is E1010. And since we already found it, we don't want to go through that long process of actually finding it again. So we can just type in E1010 right in the search bar, and it'll take you right to the part main page, or right to the part page. Find everything about it here, excitation, emission peak, all the stuff you need. So those are the kind of different ways that you can search through the part. So we showed you how to find the Excel files to search through it, how to search just knowing what type of part it is, how to search using text, and also how to search using the actual part number. So that about wraps it up for how to use the registry. Again, practice makes perfect. It can be confusing sometimes, but that's how to generally get through it. And the more you search, the more you look for parts, and the more you use the registry, honestly, the easier it gets. So once again, my name is Chris Thompson, and I'm from Pre-University iGEM team in collaboration with the iGEM Copenhagen team for Bricks of Knowledge, and hopefully you learned how to use the registry with this. So happy part hunting.